All right. Well, for my second student today, we've got Stephen Gardner. It's always a pleasure to have a student who started last year as an undergraduate, started working with me, doing some work with some toxicology with some frog embryos, and he has decided to continue on as a graduate student. So what you're getting ready to see is just hot off the presses. We started with the salamanders this January. They started breeding, and so it took like 12 days, and he's been marathoning without sleep, trying to run as many experiments as he can the last 3 to 4 weeks, because that's the only breeding season for these guys, for him to do his experiments. But he managed to get one experiment done this semester with arsenic completed with data analysis so far, and he's got like 3 or 4 still running and in the process of going. So he's doing quite well, and I will turn it over to Steve.
So, thank you everyone. I'm Stephen Gardner. Thank you for coming out despite the fact that it's horribly cold outside. So my research is using spotted salamander embryos as models for assessing environmental toxicity of arsenic. So we have the spotted salamander. Its scientific name is Ambystum immaculatum. It's a member of the mole salamander family. Basically, mole salamanders, they live underground in burrows that either they dig or that another organism has dug, and they live under detritus, fallen leaf litter, and forest floors, and they feed on invertebrate. Well, every year, as Dr. Rayburn mentioned, from January through about early March, these guys come to these types of pools. They're called vernal pools. Basically, they fill up whenever it rains in the spring, or rather the late winter, early spring. It rains, it fills up these things. The water from the surrounding environment runs off and it fills up the pools. These guys come in and they lay their eggs in egg masses, and then the adults will then go back out into the surrounding forests, and they'll live out their lives till next year when they'll come back and breed. And the developing egg masses will sit out here in this water in this vernal pool. This is actually a pool that's out here behind the hospital. It's Henry Farm Park. Basically, the eggs will stay out here and develop till they hatch about April, early May, and then they'll swim around as larvae, and they'll keep developing until the pool starts to dry up in the summer. Then they have to turn into adults, because if they don't and they're still swimming around as larvae, they'll wind up dying whenever the pool dries up. It's important to note that any kind of pesticide or metal or any organic contaminant, anything like that that's in the surrounding environment, whenever these form from the runoff water coming in from the environment, it's going to pull in with it anything that's out in the surrounding environment. So these guys, as they're developing, are going to be exposed to it. So amphibian decline, there's been a lot of populations of amphibians declining. That can be due to habitat loss, like logging, land development, or agriculture. Again, these guys, like salamanders in particular, have to have these forests in order to live, and these vernal pools in order to breed. We come in and get rid of the vernal pools and forests, then it starts, wiping them out. Pollutants like pesticides or metals, organic solvents, things like that, they're exposed to it. It can also cause harmful effects. To give you an example, the blue spotted salamander, Ambystum laterale, another member of the mole salamander family, it's endangered in Iowa, and now you can only find it in these 2 counties, Black Hawk County and Lynn County. Both of those have had reports over the last few years of them having lots of arsenic and chromium exposures and things getting released like that into the environment, so these guys can be getting affected by that. So as far as arsenic, it's already naturally in the environment in very low concentrations. Some natural activity that can cause it to actually increase in concentration in the environment could be volcanic activity or things like weathering of arsenic-containing ores. But anthropogenic sources or human interactions have caused some levels of arsenic to go up, like mining, smelting, burning coal at like power plants, or whenever they spill that coal ash from the resulting burning coal, like with the Tennessee Valley spill, it releases lots of arsenic into the environment. Arsenic is considered carcinogenic. That means it does cause cancer. It's known to interfere with DNA, cause DNA damage. The EPA has therefore set a limit on arsenic in drinking water to 10 parts per billion. That's micrograms per liter. So a very, very small amount of arsenic can be present in water, and you can be considered safe to drink it. Otherwise, you can't ingest it at all. All this kind of bearing in mind, arsenic can have adverse effects on wildlife, especially these salamanders. So my objectives in my research were to determine if spotted salamander embryos could be used as model organisms in toxicity and teratogenicity testing. Teratogen is just basically, it tells you whether or not it causes birth defects, growth abnormalities, things like that, developmental stages. And it was to determine the toxicity and teratogenicity of arsenic to developing spotted salamander embryos. So what I did is I went out to Henry Farm Park And if it had rained or the temperature, like let's say today it was really cold and then it warmed back up, that also causes the adults to come to the pond to breed. So I would go out and I would check for egg masses. And if I found any egg masses that had been laid within, preferably 24 hours, again, we're trying to see if it's causing developmental effects. So if they're already kind of older, I may have already missed the window to see if an effect would occur. So I want to get them as young as possible. And then I'll bring them back and allow them to acclimate to room temperature. I would make increasing concentrations of arsenic, beginning with the environmentally relevant 0.02 milligrams per liter parts per million and 0.2 milligrams per liter parts per million. Basically, those are some values that were reported up in Iowa, as well as from the Tennessee Valley spills, some high levels of arsenic. And higher levels of 2, 20, 200, and 2,000 parts per million. That's so that way I know for a fact I'm going to see some kind of malformation and mortality due to arsenic. I made all these up at day 0 of the experiment, the day I went and got eggs, using a 2,000 milligram per liter stock solution of sodium arsenate dibasic heptahydrate. Each concentration I made went into 4 replicate labeled plastic petri dishes, or no, not petri dishes, plastic beakers. And I also had a set of beakers containing only Phytax solution that were used as controls. Phytax solution is just ultra pure water that has only ions in it that these guys would need to live in and survive in. And it's a common solution that's used with lots of frog embryo testing for trogens and things like that, so I use those as controls. So I mentioned before that they laid these eggs out in egg masses. For my test, I had to get the eggs out of the egg mass. The egg mass itself can actually inhibit how much arsenic is actually being exposed to these guys for each embryo. I wanted to make sure they were all getting exposed to the exact same amount, so I had to get them out of the egg mass. So I gently would cut it open and remove them with a pipette and put them into a petri dish containing Phytax, and then I would look at them under a microscope. If they were developing properly, they weren't dying, they looked as they should, they went into my test. Here's a set of all my beakers set up. Once I had them in there, I kept them at 24 degrees Celsius, and each day I would come in and check for mortalities, and I would check the pH on them. Because if the pH of the solutions is too high or too low, it's too acidic or too basic, not in that order, but if it's too acidic or too basic, basically it could kill them, and I don't want to see anything from pH in my test. So I had to make sure they were kept in the pH range these guys could live in. And each day I took pictures of developmental stages, as well as recorded any mortalities, and I removed those. At 12 days, 2 days post-hatch, because they started hatching at 10 days, I went in and I took images for length measurements to see if sodium arsenate dibasic heptahydrate caused any stunting of their growth. And then I concluded the test and analyzed the data using Systat and obtained statistics with ANOVA, analysis of variance, with Bonferroni test and probe analysis. So the results, if you look at it here, this is a graph showing mortality and malformation and the response we had at different concentrations. We had 100% mortality in the 2,000 parts per million, which we expect to see. Other than that, we didn't see a lot of a higher percent of response due to mortality. It varies between about 15 to up to about 25, back down to about 15 from the 0.02s up to the 200s. And then the malformations, we didn't see a lot of malformation percent response until you get to 200, and those were about 45% malformed. Again, I'd like to note that there's no malformation for the 2000s because they were all dead, so they couldn't be malformed. The length measurements, there wasn't really any stunting of the growth occurring from arsenic due to these guys until you get to a concentration of 200 parts per million. The control lengths were 1.69 centimeters. These wound up being about 1.4, 1.49 centimeters, so it did in fact stunt their growth at 200 parts per million. So here's a picture of the controls. Here's what they look like at day 3, day 6, day 9, and day 12. By day 6, the embryo is elongated out. The gills are starting to form. You can see the eyes. Day 9, the gills are fully formed. And this is what they look like by day 12. Notice how it's straight, and I couldn't get the entire embryo in the picture because they got too big to actually fit under the camera. This is what the one exposed to arsenic at 20 parts per million looked like. Notice at day 6, it's a little more stunted than that the controls were. By day 9, the gills are still fully formed, but by day 12, this was some of the typical malformations I was seeing. Notice how it's curled in on itself a little bit more. This guy couldn't swim very well. It wasn't very active. Those are some of the malformations we're seeing at 20 parts per million. At 200 parts per million, Notice how there's a lot more malformation going on. The gills aren't really forming properly at day 6. By day 9, it's severely curled in on itself. There's some severe tail malformation. And this is one of the eyes that was more malformed at day 12. And he was actually small enough for me to, some of that stunting was going on. He was actually small enough to fit in under the microscope for me to take a picture of it. So the results, the LC50, the EC50, and the teratogenic index. The LC50 stands for the lethal concentration required to kill 50% of the embryos. That wound up being 618.99 parts per million, or milligrams per liter, with lower and upper limits of 441.17 and 989.84. The EC50 is the effective concentration required to malform 50% of the embryos, and it was 252.45 parts per million, or milligrams per liter, with the lower and upper limits of 183.77 and 420.51. And the teratogenic index, telling us how teratogenic this compound is, basically that's the LC50 divided by the EC50. Tells you how likely you are to kill something as opposed to malform it. It wound up being 2.45. For a compound to be considered teratogenic, it has to be a TI, teratogenic index, of 1.5 or greater. So it being 2.5, we do know that it's teratogenic to these developing salamanders. So in conclusion, the spotted salamander embryos can be used as models in toxicity and teratogenicity testing for environmental contaminants. The malformations that we're seeing that were caused by sodium arsenate dibasic heptahydrate include the abnormal curling that I showed, as well as tail malformations. Sodium arsenate is in fact teratogenic to developing intestinal maculatum embryos, and salamander populations could in fact be impacted by metals and metalloids in the environment like arsenic. I'd like to thank Dr. Rayburn and Dr. Klein for putting up with me every single day as I'm doing all this, and are there any questions?